different in different chromosomes. We've already talked, I think, a little bit about the fact that recombination is essential to disjunction, normal chromosome segregation. But it's turning out that actually the location of where the recombination takes place, whether it's closer to the centromere or closer to the telomeres, age, there's multiple factors. And it all doesn't get down to just the cohesin hypothesis, right? Cohesin decay. That there's pretty good evidence that decay of cohesin in older women is a contributing factor. And it's clearly that non-disjunction in oocytes are much worse than, much more frequent than non-disjunction in sperm. Recombinant, there's all kinds of things. Checkpoints in prophase 1, remember checkpoints, onset of anaphase, this is the spindle assembly checkpoint. They're actually pretty bad and permissive in females. Age-related decay of cohesin in females, because remember, as we discussed the other day, when do the female oocytes arrest in meiosis? What? Who said that?
Until fertilization.
Oh, in a woman that has a child. When did the oocytes? Let's say a 20-year-old woman has let's not think about a child. 20-year-old woman versus a 40-year-old woman. How long are her oocytes going through mitosis? No. Let's say before we're not thinking about any of them that are ovulated, the ones that are just there waiting to be ovulated. Have they arrested in a particular stage of mitosis? Meiosis. Meiosis 1.
Prophase 1.
Prophase 1 in particular. Okay. What's happening in prophase 1? Synaptonyl complex. You probably have the crossovers resolved, but we haven't had separation yet. When did that meiotic arrest? When did that arrest in meiosis 1 take place? In a 40-year-old woman or a 20-year-old woman. When they were born?
Before, yeah.
And developing what?
Like, wait, what?
When do the female germ cells arrest in meiosis 1?
Like in the very early stages, like they're developing that sort of embryo.
In the embryo, right? And remember how the germ cells migrate? You don't have to know all the details of that migration, but they're migrating from a different part, actually outside the embryo, through the gut, up the dorsal mesenteric to the gonads. About the time right after they reach the gonads, the female XX germ cells will arrest in meiosis. So a 20-year-old woman has had her oocytes, her chromosomes, held in prophase 1 for 20 years plus 9 months. 8 months. 7 months? 20 plus, including her. Okay. 40-year-old woman, 40 years plus. That's a long time for any protein complex to stay intact. If that starts decaying, you start getting non-disjunction. There's clear evidence for that in mouse, good evidence in humans, but also good evidence that's not the only explanation. So just to remind you what the cohesins are, okay, really important. Now, there's 2 questions then. I mean, that's kind of, given what we know about chromosome biology and that extended prophase 1 arrest in oocytes, that would be the first thing I think anybody would start looking at in terms of a possible mechanism. But what are the key things that we need to know about that? Is there any direct evidence for this? looking at the level of the proteins. Yes. From both mice and humans. Much more detailed in mice for practical reasons, but some in humans. But if we're thinking that cohesion decay is responsible, either that would mean there's a very low level of turnover, right? New synthesis of new cohesion that would load onto those chromosomes during the lifespan of the woman. So the alternative to turnover, though, would be, well, maybe that cohesion that was expressed and loaded onto those chromosomes in the embryo are the ones that stay. Now that's very difficult to assess in humans, but there's some beautiful experiments that have been done in mice that clearly show. Wait a minute, that's wrong. This should be, I'm sorry. It is true that the ones that are loaded, that the cohesion that's made in the embryo, I'm sorry, I'll correct that slide. The cohesion that's loaded on the chromosomes in the embryo, there is no turnover. For mice, very hard to establish. And there's some really cool genetic tricks that were done to do this. But this is, yes. Now the other thing, I just want to show you this example, and this is from mouse, just to illustrate. So, if the cohesion decay hypothesis with the age-related increase in non-disjunction is true, you would expect there to be some separation of those sister chromatids, right? Even if you're not looking for the effects of non-disjunction, you would think that if the cohesion function is reduced in older individuals, you would think you'd be able to see that at the level of the chromosomes. Now that has been looked at in humans, but again, with the limitations of humans, you'd have to get early oocytes, embryonic oocytes and look at different ages. And it has been done, but this is in mouse. Now the first thing, so the green are the chromosomes, orange are the kinetochore. The first thing you should realize, mice don't have any metacentric, submetacentric, or acrocentric. All mouse chromosomes are heliocentric. So every mouse chromosome has the kinetochore. What's the kinetochore? Yes, so that's what that is actually the spindle fiber attaches to, and the kinetochore. So basically, the kinetochore is where the centromere should be. Okay. These are young mice, real close together. These are older mice. The kinetochore is on a pair of sister chromatids in older mice, further apart. And that's, you can just look at the quantification of that. So, cohesion decay, solid evidence from mice that's contributing. Some similar evidence, but more limited from humans, but clearly other factors are involved. Okay, any questions about either anything we've covered or what you should be studying? Yes?
Is the I guess spread of the kinetochore correlated at all with like the mouse age? Like, is that proportional to humans? Like, would these mice be like 40-year-old mice?
That's an excellent point. I might not have mentioned that, but I'm glad you brought it up. An old mouse would be 2 years old.
Right.
Some strain, depending on what strain, some will make it to 3 years. It is strain specific.
So,
But 2 years, even 3 years versus 40, 50, that's an excellent point. Yeah.
I'm just wondering about like translating the models like from mice to humans.
So,
like, it's obvious that with age, but I'm wondering if like there's a cohesion decay that's more rapid in mice than in humans.
It's kind of, to a certain, so a couple of things, those are excellent questions. A couple of things you want to know, along with, is there any physical evidence at the level of the chromosomes? You'd also want to know, is there a decrease in the total amount of cohesion? Either that's loaded on the chromosomes or even in the free. That has been looked at in mice. I'm not sure if there's been enough samples to do it in humans, but there is a decay, both the total and something. Okay. The other thing, it's very clear, it's been clear for many years, decades, that older mice have a higher rate of mice than younger. So that part of the model is valid. But your question about does an old mouse equate to an older woman, especially when you're talking about decay of a protein, we have no way of knowing. One of the obvious limitations of mouse models. Any other questions? Okay. Oh. Now we're at the embryo. I think you should be able to identify these two panels, right? Zygote. Anybody remember what this is called? Let's see. That's a blastocyst. That's you, or you look like that. That little clump of cells there was you. These cells are the ones that contribute to all the cell types in the body. These cells on the outside do not contribute to adult cells. These are what form, they're important in the blastocyst stage, but they contribute to the placenta. That is what you look like a little bit later than this. This is after implantation. This is where that key event takes place. And this is literally, this is just the embryo part, the non-embryonic portion has been removed. That was been dissected off down here. This is literally just a flat, it's called the embryonic disc. It's literally, it's kind of like, think of a shell. It's literally almost flat. There's two cell layers, two layers of cells. But an amazing process called gastrulation takes place there. We're not going to talk in detail about it. We'll talk about this. Okay, this is a slide I showed you previously just to put it in context. It's about in here that germ cell development initiates, okay? That is not to scale, but here's the zygote going. What we'll be talking about in terms of when gastrulation takes place is about here, that tiny little thing, and then we go on up to here, okay? You don't have to worry about any of those. This is just another view of that, but now we see a little bit. These are not to scale with the rest of the embryos, but now you can get a little better idea. So we go just in a couple stages, a pretty short time between a 2-cell disc to something that has both anterior, posterior, dorsal, ventral, left, right patterning. And at this point, what starts forming, what happens with gastrulation, we go from having two primitive cell types that are set up in the pre-implantation embryo that differentiate into 3 cell types. And those 3 cell types are specialized and contributed to, those are called the 3 primary germ layers or 3 germ layers. Those contribute to every type of cell in the body, except germ cells. Germ cells are special. Okay, so if you want to know, if you're interested in the difference, there is by definition, a difference between embryonic and fetal, and it's not just with respect to the timing. Fetal is really when organogenesis takes place. The rudiments of all the organs are set up by late embryonic, but they're not fully developed. Okay, so the embryonic period is really with establishing each of almost all of the cell fate decisions are made, and even if you don't have the full organ, it's set up, the progenitor cells are in place in particular stages of the embryo to be able to make the heart. The heart is first, and the heart actually starts beating first. The heart, the nervous system, the limbs, okay, all that. I do want you to, all these words are in red. I want you to learn those. You don't have to know these, but we'll go through these. Okay. Fertilization to implantation. So this is where the ovary is. Here's the fallopian tube. Now, the implantation is taking place here. It's essentially a week later. Okay. Now this figure is from that assigned review, but I've added some things. here's the timing of events, 2 cell, and what's happening, 2 cell, 4 cell, 8 cell. Now, I do want you to know more than one. So, but a really fascinating thing takes place here. But the things that take place here set in motion a very important cell fate decision, and that is making the first decision between which of the cells here form this inner structure, which of the cells form that defective room. That's incredible. You know, that decision, if that decision isn't made, you don't get a viable embryo. It would not implant, and it would just be flushed out of the uterus. Okay. We've already talked about the decay of maternal RNA, just to put it in that perspective. 2 cell, but these are referred to as the cleavage stage. This is where activation of the embryonic genome. So this is putting this event or these events in context with the previous slide. Compaction is what's taking place in the memorial, and that allows the first lineage decision to take place. What happens here allows the second lineage decision. So the first lineage decision determines what makes these slightly orange cells different from these cells. Remember, both cell types are derived from undifferentiated cells in the previous step. What the second lineage decision is doesn't affect the outer cells, but it's this clump of cells differentiating into this outer kind of rim of cells. It's colored green and orange. Then implantation takes place. Question? It kind of depends. Sometimes there's, it's a real early division. Say, instead of this cell just going through one division, there might be multiple divisions, and then the embryos separate. The cleavage, they have to be at cleavage stage. The embryos separate. There are different kinds of twins. Some twins will stay in one placenta. Some identical twins will actually have 2 placentas. And some people think that if there's 2 placentas, they're not going to be identical. But from what I've read, I think 1 placenta is most common, but you can still have literally identical twins, monozygotic twins, and 2 placentas. It just depends on when the embryo divides.